Pope Aron declared that new genetic information is provided by mutations. Well, let's examine your claim, Your Highness. Okay. The human body is made up of lots of cells, okay? There are 200 different types of cells in the human body. You have bone cells, muscle cells, nerve cells, 200 different types. Each is specialized to carry out a particular function to form a particular tissue. You think this all happened by mutations, by chance, coming from an amoeba, which came from a dot of nothing exploding. There are two basic types of cells, eukaryotes, and you love to use that word. You say, boy, are humans related to whales? Yeah, they're both eukaryotes. Okay, I'm sorry. And prokaryotes, okay. All cells, whether they are prokaryote or eukaryote, have some common features. It could be maybe they got a common designer. The final count is 37.2 trillion. They asked the question, how many cells are there in a human body? 37 trillion cells. Keep in mind there are 7.5 billion people in the world. 7.5 billion. This is 37 trillion. Okay? That's what 5,000 times more cells in your body than there are people in the world. Okay. Each of the cells is very complex. This one says there are 100, let's see, that would be million, billion, trillion, 100 trillion atoms in each cell. So you got 37 trillion cells, and each one has a million, billion, 100 trillion atoms. And all this lined up, by chance, mutations over billions of years. Now, here's the human eye. Each of these lines is going down by a factor of 10. So it's 10 times smaller, 100 times smaller, 1,000 times smaller to get to a bacteria. A bacteria is 1,000 times smaller than your eye. You go all the way down here, electron orbital, atoms, small molecules. This is just a, okay, for instance, this is a dime. If you look at the eyeball of President Roosevelt, this is a cluster of little bitty cells to show how small cells are. They would fit that many in Roosevelt's eyeball on a dime. Now, the DNA strand, which we're going to talk about here, is much smaller. The thickness of a piece of paper. DNA strands are so tiny, you would have to stack 30,000 of them on top of each other, lay them on top of each other, 30,000 of them to be the thickness of a piece of paper.
That's insane. Yeah.
It has happened by chance over billions of years from an explosion. Okay. Piece of paper. An atom compared to Danny DeVito, compared to the diameter of the sun. Molecules are really complicated by themselves. There are 100 trillion atoms. They have to be arranged in a very precise order, too, for life to exist. Now, these 100 trillion atoms have to be in a real amazing order to let life exist. The DNA molecule, deoxyribonucleic acid, is unbelievably complex. If you took a ladder, a long ladder, and started twisting it and twisting it and twisting it, the ladder gets twisted and twisted, and then it overlaps itself and lays around itself like this, and then it all makes it into a chromosome. Each of these little tiny cells in your body has 46 of those chromosomes. We're talking real tiny, okay? And this will happen by chance, Mr. Nelson. You really believe that? I want to retract that statement. So let's look at a couple video clips and see the complexity of what happens when that DNA wants to make a copy of itself to make a new cell. Watch this.
These are tiny molecular machines, and they are doing this inside your body right now. To understand why, we have to zoom out. Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction, so it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body.
"46 of them in each cell and 37
You can actually see
trillion
chromosomes
cells."
under a microscope in dividing cells. Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time lapse. You can see how the chromosomes line up on the equator of the cell.
How do they know to do that?
Now, when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, as simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dynein motor. That's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction.
Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully execute their functions billions of times over inside your body at this exact instant. I'm also amazed by the scientists who were able to work out how this happens in such detail that we could create realistic depictions of them like you saw in the animations in this video. But perhaps the most amazing thing is just how much is left to be discovered, like figuring out how exactly the chromatids are pulled to opposite ends of the cell. There is still so much that we don't quite know. You know, what I find exciting is that in science fiction, for decades, we've been writing about tiny nanobots that will be injected into our bloodstreams that can heal us. But what this suggests, the existence of these tiny molecular machines inside us, it suggests that there isn't a physical limit that would prevent that. And so I think it's pretty likely that in future, we will be able to develop our own tiny molecular machines that will be able to repair our bodies better than they can repair themselves.
Regards to his new information, beneficial mutations, the thing is, a lot of them now, even if I gave him a couple, for example, it's still not going to counterbalance the damage, but a lot of it now is based on epigenetic markers, and they're used and acted to control and regulate the activity of our genes, right? So our epigenome can influence our physical appearance in much the same way as our genes do, but with no change to the underlying genetic code. For example, diet alters our teeth, swimming alters our spleen size, high altitude alters our chromosomes, hard work alters our bones. But the thing is, there's no large scale evolution, no new information is added, just expressed.
You want to pontificate. You said, new genetic information is provided by mutations. That's like saying, if we have enough mistakes in this factory that makes cars, it'll all of a sudden start producing airplanes. Now you just breezed through this with your 10 or 12 or 15 reasons, whatever it was, that I'm wrong and you're right. I'm going to go through them one by one as I get time, because I'm sorry, son, you're a low priority for me. But I'm going to answer them because you're affecting other people. You are incorrect, you are mistaken, or you're deliberately lying, okay? You are pontificating. It is not true that new genetic information is provided by mutations. You are wrong, I am right. It is not provided. There is none. An example of you pontificating without evidence or authority. So, if you can demonstrate that new genetic information is provided by mutations, you should be honest enough to admit to your disciples that, number one, there's only a few, maybe 5 or 10, that have been even claimed of the trillions you would need. It would take quintillions of beneficial ones to change an amoeba to a whale or a hummingbird. Quintillions, sextillions, octillion, novillion, decillion. 99% of mutations are harmful, fatal, or neutral. You should admit this to your followers, okay? If not, I'll admit it for you. Folks, this is what he's not wanting to tell you. Wake up, okay? He's leading you down the wrong path. So let's see. Science Daily listed 0. BiologyWise website had 5 supposed beneficial mutations. Physics.org had 1. Study.com had 3. Berkeley had 2. University of Cambridge had 2. Sanger Institute 1. Charles Darwin none. Where are the beneficial mutations? Even Mr. Nelson, if I said, okay, all of them are true, I'll give you 10 beneficial mutations. You know how many changes there would have to be to go from
Trillions.
quadrillions, quintillions, sextillions, and you can find maybe 10? So a change in an organism's DNA can cause changes in all aspects of its life. Mutations are essential. This is from Berkeley, okay? Essential to evolution. They are the raw material of genetic variation. Without mutation, evolution could not occur. Okay, I would agree. And mutations are all harmful, so evolution does not occur.